cells is called callus. After that, if we add some auxins and cytokinins to this medium, it results in the formation of shoots or roots. Observe here, in this shoots and roots are formed. Differentiation cells. For example, if we add growth regulators like high auxin, and low cytokinins, results root formation. Root formation. Or with low auxin and high cytokinins, results in the formation of shoot. Auxins equal to cytokinins, results in the formation of callus. This is about growth regulators and forms like shoot and root. The formation of root and shoot is called organogenesis. And let us see the process of organ culture. How the organ culture can done. Out of this, I have taken anther culture. I have taken anther in a medium and the pollen mother cell. The pollen mother cells growing meiosis division and becomes a haploid embryo, then transferred to then it becomes to a complete individual haploid plant. This is about anther culture and it is called it as organ culture. Suppose if we lost any finger or any body part of a organ, can we regain it? No. So, our body cells are unable to produce a complete or individual organ, but it is the only capacity of the plants to grow into a individual plant. If we take any part of a plant and grown into a complete individual plant, it is the only capacity of the plant is called totipotency. Totipotency was first coined by Morgan in 1901. S.T. Stewart first experimentally demonstrated totipotency from root secondary phloem tissue. This is about totipotency. Now, let us see tissue culture techniques in an chart. See here, observe here, I have selected a plant, the plant with desirable characters, and we have selected explant. Explant is nothing but any part of a plant, that any part of a desirable plant is called explant. The explant may be a axillary bud, root tip, shoot tip, bud, etc. And it must be sterilized with detergents to clean the surface of the leaf. After that, it must be washed with sodium hypochlorite to kill the microorganisms do not grow along with this explant. Now the explant is sterilized explant. After that, in a medium, it is a medium. Now in a medium it consists of nutrients, and it was nutrients like vitamins, proteins, amino acids, etc. In this now sterilized explant is inoculated into this sterilized nutrient medium, and it results in the formation of callus. This is a callus. Callus is nothing but a group of cells. Sometimes a callus may directly develop into a embryo like structures. It is called somatic embryoids or embryoids, and results in the formation of plantlets and then transferred to pot plants. Pot plants means like it must be kept in 2 to 3 weeks in a glass house, and then slowly transferred to hardening and transferred to fields. This is about tissue culture technique. This is about tissue culture technique. Let us see. Tissue culture technique process, let us see in the form of a flow chart. Preparation of medium. Just now we have prepared a medium by adding some agar-agar to it, and it must be mediums must be taken in a test tube, conical flask, etc. and explant, and then into the explant must be the sterilized explant must be inoculated into the sterilized nutrient medium. It is called inoculation. After inoculation, we must put the plant placed in a incubator for 2 to 3 weeks, and it results in the formation. After 2 to 3 weeks, we can observe callus like formation, and this transferred to, and if we add to callus like growth regulators like auxins and cytokines, results in the formation of root and shoot, and the formation of root and shoot is called organogenesis, and then slowly develops like a plantlets. There is another way, somatic embryoids, and the somatic embryoids must be, then they can be encapsulated with sodium alginate to produce synthetic or artificial seeds, and then transferred to medium, and then it develops into a plantlets, and after this artificial seed preparation, then slowly develops like plantlets, and then transferred to the fields. This is about tissue culture process. Let us recapitulate. The formation of shoot and the root is called organogenesis. Here see how the how the individual plant is raised from a single plant. Yes. Observe here, there are different types of culturing plants. Plant culture tissues, the steps. First we have selected a healthy vigorous plant that must be with sterile desirable characters and transfer, that may be sterilized, that must be sterilized with detergents. Second step, the explant must be encouraged to divide the cells and encourages cells division. It may be solid or liquid. Second step, sometimes the particular medium requires, we must be established by trial and error. Multiplication. The cells of explant can grow in forms a mass of loosely arranged cells which is manipulated by varying sugars contents. We added some growth regulators like auxins and cytokinins, results in the formation of shoots. Here the auxin is low and the cytokinins is high. So, it results in the formation of shoots. Hello?
Hello, my name is Prashant from Dampata Institution.
Good afternoon, Svero.
Good afternoon.
What is your doubt?
Take place in our institution. How much time to take explant?
Again repeat the question.
How much time taken?
For which purpose?
Explant?
Only 1 month it takes time and complete individual plant can be raised. From a single cell, we can complete individual plant is raised within 1 month or 2 months and then transferred to fields. Hello?
Hello?
Hello, Svero.
Hello, good afternoon, Svero.
Good afternoon.
Hello?
What is your doubt?
This is J. Kinesh from TSWREIS, Chilkur, Svero.
Sir, Svero, I not understand.
Svero, what is the use of tissue culture, Svero?
What is tissue culture?
What is the use of tissue culture?
We can, in it, tissue culture advantages also is there. Tissue culture uses are very more. In a short time, we can grow more number of plants in a limited space. Here the population is increasing day by day and the food production is not sufficient for the growing population. So, the tissue culture is used and we can in a very short time period we can produce more number of plants. Thank you. Thank you, Svero. See, multiplication of explant. The explant can give rise a callus. Callus is nothing but a mass of cells. And the callus may be subdivided into a number of times. Sometimes a callus may develop into a embryo-like structures and the embryo is made directly used for culturing. Hello? Hello?
Hello.
Hello, Svero.
Good afternoon, friend. Auntie Swapna Svaro from Wider Kammadanam district.
Tell me, Svero, what is your doubt?
Your class is nice, friend.
Thank you, Svero.
What is the use of cytokines? What is the use of cytokines? Can you tell me?
Cytokines results in the formation of root and shoot. And it results in the formation of organogenesis and it produces a complete individual plant. By adding cytokinins, we can produce shoot-like structures. Now, let us recapitulate once again. Root formation. The rooted shoots and potted up and hardening are gradually decreasing the humidity. Here see so many of plants in a very short period in a very limited space. Here the culturing is done. See how the plants is transferred to pot plants. The soil which is used for culturing is raw. Here we are taken a plant and a plant must and the explant is taken a mass. Hello?
Hello. Hi, Svero.
Hi, Svero.
I'm Mansa from Marikonda Magnet School.
Tell me, Svero, what is your doubt?
How can we culture virus plants from culturing medicine? How can we produce virus-free plants?
by
By
repeat again.
how can we produce virus plants? So by culturing medicine.
And we have selected a healthy vigorous plant na, a virus free plants. That's why we have selected a desirable plants and with virus free plants, and we have to culture into a medium. Plant tissue culture. Here we have taken a root, a carrot, and we taken a shoot from a carrot, and this is explant. Then it is transferred to a sterilized nutrient medium, and it forms like a callus, and then it is the callus may directly develop into a embryo like structures, that is called embryoids, and it is a forming like a embryos, and then slowly transferred into a plantlets. This is also one type of tissue culture. Here you see we have taken a explant and then slowly transferred into a sterilized nutrient medium, and then into a parts, and then transfer into a fields. Here the plant is disease free plants. A more recent advance in the use of the tissue culture along with.
Hello, Swaro.
Hello, Swaro. Tell me your doubt.
Good afternoon, Swaro.
Good afternoon.
I'll ask you one question.
Tell me, Swaro.
What is meant by totipotency?
Totipotency means nothing but any part of a plant, and that can develop into a complete individual plant is called totipotency. Totipotency this is the only capacity of the plants could produce into a complete individual plant. This is about totipotency. Thank you, Swaro. Anther culture. For anther culture we have taken pollen, and we can culture, and the individual plant is haploid plant, because we have taken a anther, that is why it is haploid plant. Production of virus free gram. Protoplast cultures. Shoot culture, and we have taken here shoot tip, and then it culture into a complete individual plant. Micropropagation means by applying this technique it is possible to produce a large number of plants in a very short time, and it gives rise to a complete individual plant. This is about micropropagation. Stomaconal variations. It is a hybrid of potato and tomato. It results in the formation of pomato variations. Like root heads are potato and the leaves like tomato. It is called, and it is failed. Advantages for tissue culture. Using synthetic seed technology. Synthetic seeds are directly used in fields, and fruits, like every part of a plant we can use it. Applications of the medicine and research. There are various applications of tissue culture in a wide array of scientific industries, cancer cells and skin grafting. Advantages of tissue culture. A single explant can be multiplied into several thousands of plants in less than a year. This allows the fast commercial propagation of new cultivators. Taking an explant does not usually destroy the mother plant, so rare and endangered plants can be cloned safely. and the plant must be selected with healthy vigorous plant with virus free plants, and we for plant tissue cultures also there are tissue banks. Plant tissue cultures are approved media, easier to export than in soil grown plants and can pathogen free, taken up little space and export in a culture. Tissue culture allows fast selection for crop improvement, explant or chosen superior plants then cloned. Tissue culture clones true to type. True to type means here if I have taken any explant of any part of a plant and it grows into a complete individual plant as it is as an explant. That is why it is call it as true to type. Disadvantages. There is no disadvantages, but it is a technique. It can be followed by only, it cannot be followed by farmers. That is why it is, they think it is not done for our illiterates. Cost product is also more. Finally, to conclude, our topic is tissue culture. It is the best way for the propagation, various strategies for production of food production on par with the population. I am very thankful to our secretary sir and our principal madam who behind me and full support and give a full support to us. Especially thank for our botany madam Lakshmi Narasimha madam and Deborah madam who helped me a lot, and I am thank you very much, and I help those who helped for charts like Divya and Saujanya, Nimisha, they helped for me a lot in preparing charts for Mana TV. Thank you for giving this opportunity. Have a nice day.